Teaming with Microbes, revised edition. Jeff Lowenfels, Wade Lewis. Chapter 4 Archaea. Just a few years ago, no one would have thought to include Archaea in a book on the soil food web. Uh, these microorganisms, at first considered a weird and unusual subset of bacteria, were known to live only in extreme environments, such as around geysers or in hot water ocean vents. Since these spots don't exactly feature in agricultural or horticultural areas, Archaea were not considered soil food web members. Then, at the turn of this century, directly as a result of advances in genetic identification of microbes, Archaea were found in soil. Moreover, it appears that the role they play in nitrogen fixing, taking atmospheric nitrogen unavailable to plants and converting it into plant-usable form, is a crucial one. Now they have our attention. Backstory. Archaea were first identified in the early 1970s by Carl Woese, W-O-E-S-E, Carl Woese, and his research associate George Fox, who were studying bacteria at the University of Illinois. Their interesting conclusion: bacteria could be divided into two distinct groups, normal bacteria and a new second group of extremophiles that not only survive but thrive in high temperature environments. The second group produced methane, like the bacteria in the large intestines of humans, but most important, these new bacteria had a distinct genetic makeup. It was shortly found that not only was the genetic makeup of all Archaea bacteria, as Woese first called them, different from other bacteria, it also differed greatly from the genetics of eukaryotes, organisms possessing cellular membranes, distinct nuclei, and cytoskeletons, be they protists, fungi, plants, or animals. Bacteria, quote-unquote, was dropped from the name altogether. Uncovering a new form of life so late in the history of biology was a stunning development. It led Woese to draw a new tree of life with three main domains, or kingdoms: bacteria, archaea, and eukaryotes. Many believe that the kingdom archaea is the oldest of the three branches, since the conditions in which many archaea live are thought to be representative of the extreme conditions that existed during the establishment of life on Earth. Morning. Mitochondria probably evolved from cyanobacteria. Right. Oh. Okay, so we have the tree right here, actually. The Tree of Life, illustration by Nicole Rager Fuller. Oh gosh, you can see on the left here, it's got the bacteria. This rectangle covers the rest of the archaea here in the center, and then the eukaryotes. Oh man. Bacteria, Euryarchaeota, thermo, soil, marine, Prenarchaeota, and then eukaryotes, like the protists, the fungi, the plants, and the animals. Ooh. Very interesting. Bacterial lookalikes. On the matter of distinguishing between archaea and bacteria, the word impossible comes to mind. It doesn't help that they are the same size. That is to say, really small. As with bacteria, as many as 500,000 archaea can fit inside the period that ends at this sentence. Most archaea and bacteria look alike, so much so that using the eye, even at high magnification, doesn't reveal any differences. Like bacteria, archaea usually travel by whipping about one or more flagella. And when they are not traveling, archaea too can form large colonies, though some archaea live a more solitary life, as do some bacteria. Archaea grow and multiply, just like bacteria. They divide. One study suggests archaean cells divide every 20 minutes in ideal conditions. You don't have to do the math to know that in a short time the world would be a very different place if ideal conditions existed. Archaea often exhibit the same three basic shapes as bacteria, though those in the salt-loving genus Holoquadratum are square, a shape not seen in bacteria. Um, these are also needle-like filament archaea. Oh, sorry. There are also needle-like filament archaea that look similar to actinomycetes. Some form perfectly rectangular rods, and some archaeans, to use the alternative Trekkie plural form for them, are irregularly lobe-shaped, triangular, and even shaped like teacups. What makes archaea unique? With so much similarity, why did Woese find reason not only to distinguish archaea from bacteria, but to declare them a whole new kingdom, a separate branch on the tree of life? To begin with, although both bacteria and archaea have cell walls that hold in and protect the material inside, the cellular membranes of archaea contain the cellular membranes of archaea contain ether lipids, while bacterial cell walls do not. Archaea walls contain amino acids and sugars that differ from those found in the cell walls of bacteria. And unlike eukaryotes, archaea cell walls never contain chitin, as do those of fungi. Nor do they contain cellulose, as do the cell walls of plants. This is complicated stuff, which harkens one of us back to college organic chemistry. The reason he is now... This is complicated stuff, which harkens one of us back to college organic chemistry. The reason he is not now a physician. Obviously, it takes computers and analysis well beyond the capabilities of the gardener to discern these differences. Trust us, as we do, Carl Woese and his colleagues. On a cellular level, these distinctions are so important, they are sufficient not only to split these weird, sometimes oddly shaped organisms into their own group, but to put archaeans into their own kingdom. The second characteristic that separates archaea from bacteria has to do with genetics. Both archaea and bacteria are prokaryotes. Their genetic material is not enclosed in a nucleus. However, the genes of archaea, but not bacteria, are more closely related to the genes of eukaryotes, those organisms whose DNA is enclosed within a nucleus. Specifically, in both archaea and eukaryotes, similar enzymes are involved in the synthesis of ribonucleic acid, or RNA, which itself is a key to the synthesis of proteins. Another set of characteristics sets archaea apart from both bacteria and eukaryotes. Archaea use many different substances for energy: sugars, ammonia, hydrogen, and many metal ions. Some can even use sunlight as a source of energy. Others use carbon dioxide. And while some bacteria and eukaryotes can reproduce by spores, Archaea don't have this ability. Finally, if you really want to get down to detail and differences between archaea and bacteria, while their mode of transport is the same, flailing flagella, the chemical composition of the flagella and the mechanism that makes them operate are different. Talk about splitting hairs! Types of archaea. Determining the difference between a bacterium and an archaeon then requires work on a genetic level. We think of it just like reading barcodes. Each organism has a unique code. Using DNA and RNA sequencing, scientists can read the genetic makeup of a given sample, the barcode, just like the checkout clerk at a grocery store. These techniques work on DNA found in liquids, solids, and gases. Thus, all sorts of environments can be assessed, including soils. So far, approximately 250 different kinds of archaea have been identified. More will be found now that scientists know what they are looking for and have the proper tools to do the job. Archaea play a big role in the carbon cycle, and one way to group them is to look at how they get their energy and nutrients. Some archaea are photoautotrophic, containing energy from light and using CO2 as their source of carbon. Others are chemoautotrophic, using inorganic chemicals as their energy source while still employing CO2 as a source of carbon. Yet others are photoheterotrophic, which means they use light for energy but organic chemicals to obtain carbon. Finally, some archaea are chemoheterotrophic, using organic chemicals for energy source as well as for carbon. Archaea are found in hot springs and salt lakes, living in rock buried deep in the earth under miles of ice, and in the hot deserts, environments inhospitable to most other living organisms. These extremophiles are able to survive in environments because they have these extremophiles are able to survive such environments because they have developed specialized enzymes that work at high or low temperatures, and because their proteins can fold tightly to avoid destruction by salt, heat, or cold. Special characteristics of their cell membranes also help make them resistant to heat. Archaeons that live in these habitats don't need oxygen. They are all anaerobic. Oh. Some archaea, known as acidophiles, have made cellular and metabolic adjustments so that they can live in extremely acidic environments with a pH as low as 1. They constantly pump hydrogen ions out of their cells, maintaining a much higher and only slightly acidic internal pH of 6.5. Another group of archaea are classified as halophiles, or salt lovers. Thermophilic archaea live in high temperatures, and the cold-loving or psychrophilic archaea thrive in frozen areas. Many of these extremophiles are also methanogens. Their metabolic activities produce methane. It may be a surprise to learn that smelly swamp gas and cattle flatulence, heretofore thought to be produced by bacteria, are actually the byproduct of archaea going about their daily activities. Methanogenic archaea are found throughout the oceans. In fact, so many of these organisms thrive in ocean plankton. It is now estimated that the combination of archaea in the ocean and those in soils makes these microorganisms the most abundant life form on Earth. Harnessing the properties that enable archaea to survive harsh and inhospitable conditions could have great economic consequences. But techniques for culturing archaea are still in their infancy. We are certain this and other difficulties will be overcome, and archaea will become important biotech tools. Archaea and nitrogen cycle. Sorry. Archaea and the nitrogen cycle. To bring all this back to the subject at hand, it turns out that archaea also participate in the nitrogen cycle depicted on page 48. This discovery was made in two steps. First, scientists isolated from ocean-dwelling archaea the specific gene that produces the enzyme needed to oxidize ammonia. When they looked for the same gene in soil samples, they discovered it was a whopping 3,000 times more prevalent than the counterpart gene in bacteria. They also looked at the amounts of lipids associated with the organisms in the soil and confirmed the presence, and, more astounding, the dominance of archaea in the nitrogen-fixing processes in the soil. Even more interesting, the greater numbers of archaea continued at deeper soil depths, whereas the bacteria count petered out with depth. Samples taken around the world showed the same results, leading many to conclude that archaea, specifically the Crina archaeota, are the most abundant ammonia oxidizers in soil. This discovery is an important one, if for no other reason than nitrogen gases include nitric and nitrous gases. These are greenhouse gases, and it makes sense to figure out what proportion of them may be traced to the activity of archaea. There is still so much to learn about the size and scope of the role archaea play in the nitrogen cycle. How much ammonia oxidation is from archaea, and how much is from bacteria, for example. No matter how big their part, archaea have important implications for the gardener who relies on the soil food web, or teams with it to provide nitrogen to plants. Decomposers. Archaea, like bacteria, are decomposers. They break down organic and inorganic material, recycling elements needed for plant life. Some archaea get their nutrients by oxidizing sulfur compounds, which they can release from rocks. These archaea have been found there where there is volcanic activity, such as Yellowstone National Park and Mount St. Helens in the United States, in thermal areas of Iceland, Russia, and Japan. They are also abundant along sea floor vents where they either oxidize sulfur or use sulfur to oxidize carbon compounds. Either way, sulfur, a major plant nutrient, is freed and made available to other organisms in the web of life and ultimately to plants. Methane is much more efficient at trapping heat in the atmosphere than is the more infamous CO2, and we believe should be a greater cause of concern. It used to be that bacteria got the blame for methane. No longer. When certain archaea break down organic matter, they not only make nitrogen available to plants, they produce methane, a key component to greenhouse gas. One large source of archaea-created atmospheric methane is rice production. Methanogenic archaea decompose matter in flooded rice fields, resulting in both plant-usable nitrogen and methane. Given the scale of rice farming around the globe, this is a serious contribution. Methane levels have increased by about 150% since 1750, and it has been suggested that 10 to 25% of the methane emissions worldwide come from the activity of methanogenic archaea, a tremendous, significant amount. This group of archaea is, not surprisingly, under intense study by scientists trying to figure out ways to lower the amount of methane produced in agricultural activities. Most gardeners are now aware of the hypoxic or oxygen depleted dead zones that form where agricultural runoff accumulates. Places like the waters at the mouth of the Mississippi River in the Gulf of Mexico. The nitrogen and phosphorus in this runoff allow algae and cyanobacteria to proliferate and thrive. They use up the oxygen and what happily does not require oxygen and is standing by to decompose these organisms, anaerobic archaea. Oh boy. Other benefits of archaea. Yeah. Any dairy farm near a river, absolutely. Any dairy farm near a goddamn water table. Yeah. Certain archaea are responsible for the production of archaeo... Oh, shit. Let me try and read that. Archaeosins? Archaeosines? Archaeosins? Yeah. Certain archaea are responsible for the production of archaeosins, an entirely new class of antibiotics. Several have been identified and even manufactured, and the assumption is that there are hundreds more. The structure of these antibiotics is different from those produced by bacteria, so they act in ways existing antibiotics cannot. That there do not appear to be any pathogenic archaeans makes for interesting possibilities. Perhaps they can be used against pathogens, or both plants. Perhaps they can be used against pathogens of both plants and humans that have become resistant to antibiotics produced by bacteria. Archaea are already an important source of specialized enzymes that work at high temperatures. One of these figures in the cloning of DNA. Others are useful for making organic compounds. Sewage treatment plants use archaea for purification, and the mineral industry puts them to use in some refining processes. Archaea enzymes are even included in some modern laundry detergents. Hot water rinse, anyone? Oh. Clearly there will be applications using archaea or their byproducts for gardening. And there is always the possibility that archaea will provide nitrogen in artificial soil, food webs, established in such extreme environments as the moon or Mars. Yeah. Archaea, like bacteria and fungi, form relationships with other members of the soil food web. One such interaction is between methanogenic archaea and protozoa in the digestive tracts of ruminants, termites, and other animals that consume cellulose. The protozoa break down the cellulose and release hydrogen, and methanogenic archaea convert the hydrogen to methane during their metabolic process. Even some protozoa are host to these types of archaea, and the human large intestine contains archaea that aid in digestion, working mutually with bacteria that also live in the gut. Non-methanogenic archaea have been found in the soil of boreal forests, but we wait further study to learn about their metabolic activity and other characteristics. As for the soil in your yard and garden, it is colonized by archaea, which inhabit the carbon-rich rhizosphere immediately around the roots of the plants. Since little else is known about the specific roles of these soil-based archaea, we will use in this book the admittedly improper bacteria instead of bacteria/archaea in future references. We mean no disrespect to the archaea, whose presence we dutifully acknowledge, and don't mean to suggest they are inferior organisms to bacteria, but without genetic identification abilities, we can't tell them apart. There is more to come. The discoveries continue, fully understanding the impact of archaea on the production of plant-usable nitrogen, the role they play in the creation of methane, and the scope of their interaction with other members of the soil food web will all help further the understanding of what goes on in the soil to help plants grow. Obviously, though, Archaea are important members of the soil food web and can no longer be ignored. Oh. Interesting. So there was chapter 4. Oh. Oh. Oh, ho, 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 yes. Oh, gosh. That's so funny. Man, my phone's going off like crazy right now. Hold on. Alright, sorry. I was watching a clip of me getting murdered and everyone walking away in Among Us. Yeah. Oh, no worries. Oh, no, no, no. I would be more so a guest speaker. Yep. Yes, yes. Welcome, welcome. What did we just cover? We talked a little bit about the tree of life for bacteria. There's the oh gosh, the archaea. There's bacteria, archaea, and eukaryotes. Uh, while they all have these mad distinctions, it has been extremely difficult for us to like identify them until they get to the level of like taking a sample to the lab and getting DNA and RNA sequencing and testing and shit, right? Yep, it is the most arcane of bacteria, they believe. Yep. An extremophile, capable of surviving in very harsh climates, found mostly in places like around volcanic vents, in the ocean. Uh, they were talking about finding them deep underground in the ice, near the ice with Russia. The geysers that we have in Yellowstone National Park. And they are a contributor to the greenhouse gases that are happening. They make the methane. They're decomposers in the soil, converting stuff into the methane is what we were discussing. At first, they thought it was bacteria that was making the manure poopy smell that you smell of the cow poop and fertilizer, and it was really the archaea. If I regurgitated that properly, I'm sorry. Yeah. It was very interesting. Yeah. They believe that there is potential for good use with the archaea, but they don't have the means at the moment to really put them towards crazy things. They were just saying this thing about how they're using archaea enzymes for the processing of minerals. Yeah. Sewage wasteman. Sewage treatment plants are using archaea enzymes to treat their facilities for purification, as it described in the book. Yeah. Yep. But it's the freaking the archaea are there. That's what it was describing, was that the archaea are also in our guts. So it's the archaea working hand in hand in the guts of the cows that are producing the methane that the cows burp. If I read that correctly, right? Yeah, the archaea are all over. It was saying, it was like, we. It's just, it was something that they were able to identify them specifically because they had seen them in these other environments. But then like they were able to do improved testing, the DNA sequencing and taking samples, and they found out that it was like also in the soil and stuff. It's just something that they weren't able to identify at first with the thing. Mm-hmm. Mm-hmm. God damn Archaea. Mm-hmm. Oh yeah, that's how it sounded out. It's Archea, I think. Wait, sorry. Let me go back to it. Sorry. Archaea. Archaea. Yeah. Oh man. All right. Sorry, I'm gonna have a quick bowl before I do the second chapter here. Thank you for hearing me out while I read the book. I'm sorry if I botched anything. Uh, yep. So far, we had learned about what was it? The soil food web at a glance. Soil conditions. The different parts of the soil, what make up soil, and bacteria. Looking at bacteria a little more closely. Chapter 5 here is fungi. Yeah. Mhm. It's pretty cool to learn. I mean, like I've learned that about how the plant's not able to build up defenses naturally with the microorganisms in the soil to fend off all sorts of things. When you're just relying on like solutions in salt or liquid based solutions, instead of relying on the soil itself. You're feeding the plant and it just goes through. The plant catches what it wants and then it's gone. It doesn't stay trapped. The having the microorganisms in the soil, you have this whole process going on where like the bacteria and the fungi are breaking things down, recycling it. The exudates are being secreted. They're establishing colonies around in the soil, around the roots, the bonds with the roots and the microorganisms in the soil, the mycorrhizae association, and other means in which all these plants are all these things are producing nitrogen, nitrates, converting ammonium into nitrates, or like we just learned about archaea, taking that shit and making it into methane. It's crazy. Yeah. So like all of that active and alive in the soil and having a great relationship fends off all that other stuff. Mm-hmm. Yeah. And then it's plant specific with some things. Yeah, just depending on what you want to grow. And like with cannabis, it's that we were talking about those ratios. Yeah. Yep. Mm-hmm. Like if you're growing a banana tree indoors, replicate the environment that the banana tree is growing outdoors very well. You know, tropical climate, humidity. What kind of stuff is there? Oh, yeah. I've taken rocks and thrown them from one end of the room to the other and called it time travel. Would you say the same thing for a plant on wheels? Alright, backdoor eating. Sorry. Teaming with Microbes by Jeff Lowenfels and Wayne Lewis. Chapter 5 Fungi. Over 100,000 different kinds of fungi are known, and some authorities suggest 1 million more are out there waiting to be discovered. Say the word, however, and most gardeners immediately think of the familiar white toadstools, bracket and coral fungi, and puffballs that appear in the lawn or on the bark of trees. Or they know soil fungi from the disease they cause. More on this later in the chapter. But except for the white threads and the spore-producing mushrooms, soil fungi are as invisible as bacteria, requiring a microscope of several hundred power to be seen. Even the visible congregations of mycelia are usually hidden in the organic matter they are in the process of decaying. Fungi, too, are underappreciated by gardeners, and yet they play a key role in the soil food web and are an important tool for those who garden using soil food web principles. It wasn't too long ago that they were considered plants without chlorophyll, and included for classification purposes in the plant kingdom. But because fungi are unable to photosynthesize and build their cell walls from chitin instead of cellulose, among other unique characteristics, they are now placed in their own kingdom, in the domain Eukarya. Fungi, like higher plants and animals, are eukaryotes, organisms that have cells with distinct enclosed nuclei. Each cell can have more than one nucleus. Fungi usually grow from spores into thread-like structures called hyphae, singular hypha. A single hyphal strand is divided into cell walls, or septa, singular septum. The walls connecting hyphae cells are seldom completely sealed off from other cells in the strand, thus allowing liquids to flow between cells. Masses of invisible hyphae growing in close enough proximity form visible threads, or mycelia, singular mycelium, which you may have seen in decomposing leaf litter. Fungi reproduce in many different ways, not just by spores, but never by seeds, as the most advanced plants commonly do. Yeah. A fungal hypha is considerably larger than a bacterium, the average length being 2 to 15 micrometers with a diameter of 0.2 to 3.5 micrometers. Still so thin that it takes hundreds of thousands of individual hyphal strands to form a network thick enough for the human eye to see. A teaspoon of good garden soil may contain several yards of fungal hyphae, invisible to the naked eye. Millions upon millions merge to produce something as obvious as a king bullet or an intricate Amanita muscaria in all their fruiting glory. These and other mushrooms are simply the fruiting bodies of fungi. Consider the energy and nutrients required to produce them. One major advantage fungi have over bacteria, and perhaps the reason they were misclassified for so long as plants, is the ability for fungal hyphae to grow in length. Unlike bacterial cells, whose world is a very finite one, fungal hyphae can travel over space measured in feet or meters, distances that for a bacterium are truly epic. And unlike bacteria, fungi do not need a film of water in order to spread through the soil. Fungal hyphae are thus able to bridge gaps and go short distances, which allow them to locate new food sources and transport nutrients from one location to another, relatively far away from its origin. The ability to transport nutrients is another key difference between fungi and bacteria. Fungal hyphae contain cytoplasm, a liquid circulated throughout the septa in their cells. When a hyphal tip invades a nematode, for example, it drains its hapless victim of its nutrients and distributes them in the hyphal cytoplasm and from there through the main body of the fungus? That's brutal. Sorry. Oh, that one. The Amanita muscaria. Oh, that's cool. Sorry, I was reading that. I was very fascinated. Sorry. Let me read it again, because it's fucking brutal. When a hyphal tip invades a nematode, for example, it drains its hapless victim of its nutrients and distributes them in the hyphal cytoplasm, and from there, through the main body of the fungus. Nutrients are thus transferred from the tip of the fungal hypha to a wholly new location that could be several yards away. Think conveyor belt. Once inside the fungus, the nutrients are immobilized and will not be lost from the soil. Yeah. Oh my. Okay, so we were talking about there's a diagram of a hypha, the fungal hyphae, the long strands of the fungus. And then I was talking earlier about the septa, the septum. It's like the bridge between those little bits of the fungus. You see the nucleus there in the center? Yep. Oh yeah, and then they were showing the same pictures that freaking tree herder posted too. There's the Amanita. And then what's the other picture? Hold on, I'm sorry. Fungal spores are produced in bodies that rise above the fungus to help dispersal. Yep. A close-up of fungal spores. There you go. Fungi produce special structures. For example, mushrooms above ground or truffles below, to disperse spores. Since fungi grow in all sorts of environments, they have devised some elaborate methods to achieve spore dispersal, including attractive scents, triggers, springs, and jet propulsion systems, to ensure survival. Fungal spores can develop tough membranes that allow them to go dormant for years in the conditions, if the conditions are not right for immediate germination. As with bacteria, fungi occur universally. Some species even exist in the frozen region of Antarctica. Airborne dispersal of spores explains why visitors from, say, Alaska will recognize species of fungi growing in far-off Australia. While dormant spores can be found around the world, they need the right conditions to germinate and grow. Thus, fungal spores may be found continents away from their source, but they may not be functional because the conditions for growth are not right. Fungal growth and decay. While some fungi prefer the softer, easier to digest sugar characteristics of the foods that feed bacteria, most go for the tougher to digest foods, mainly because bacteria are better and faster at grabbing and taking up the simple sugars. Fungi, however, win in the competition for more complex foods. They produce phenol oxidase, a strong enzyme that dissolves even lignin, the woody compound that binds and protects cellulose. Another characteristic of fungi is their ability to penetrate hard surfaces. Fungi have perfected apical growth. That is, growth at their hyphal tip. Apical or tip growth is an incredibly complex process, an engineering job akin to building a tunnel under a river, and requiring great coordination of events. Holy shit! I'm gonna reread that. Another characteristic of fungi is their ability to penetrate hard surfaces. Fungi have perfected apical growth, that is, growth at their hyphal tip. Apical or tip growth is an incredibly complex process, an engineering job akin to building a tunnel under a river and requiring great coordination of events. Even before electron microscopes, scientists identified a dark spot, the Spitzenkorper, in the tip of actively growing hyphae. When hyphal growth stopped, the Spitzenkorper disappeared. It seems this mysterious region has something to do with controlling or perhaps directing apical growth. During apical growth, new cells are constantly being pushed into the tip and along the sidewalls, elongating the hyphal tube. Materials for the growth of fungal hyphae are supplied to the advancing tip by the cytoplasm, which transports vesicles loaded with all the necessary construction supplies. Of course, it is important to keep extraneous material from flowing into the hypha, as well as out. Of course, it is important to keep extraneous material from flowing into the hypha, as well as out while this growth is happening. All the while, powerful enzymes capable of dissolving all but the most recalcitrant carbon compounds are released as the new cells are put into place. Yeah. The book, have you checked the title of the stream as well? If you guys check the title of the stream, you could see what's going on in the stream sometimes on Twitch. Not trying to be insulting, but it is kind of insulting to us when you don't check that out before looking into that stuff. Yeah. Back to reading. Yeah. Okay. Let me read that again. Of course, it is important to keep extraneous material from flowing into the hypha as well as out while this growth is happening. All the while, powerful enzymes capable of dissolving all but the most recalcitrant carbon compounds are released as the new cells are put into place. Think about it. These enzymes are powerful enough to convert lignin, cellulose, and other tough organic material into simple sugars and amino acids. Yet they do not decay the chitin cell walls of the fungi. Fungi can grow up to 40 micrometers a minute. Discount for the moment the speed, which is incredibly fast for such tiny organisms, and compare the distance covered to the moment of a typical soil bacterium, which may travel only 6 micrometers in its entire life. As with the death of any organism in the soil, the death of fungi means the nutrients contained within them become available to other members of the soil food web. But when fungi die, their hyphae leave a subway system of microscopic tunnels up to 10 micrometers in diameter, through which air, water can flow. These tubes are also important safety zones for bacteria trying to elude protozoa. Protozoa are considerably bigger than the tunnels. Yeah. Fungi are the primary decay agents in the soil food web. The enzymes they release allow fungi to penetrate not only the lignin and cellulose in plants, dead or alive, but also the hard chitin shells of insects, the bones of animals, and as many gardeners have learned, even the protein of strong toenails and fingernails. Bacteria can hold their own, but they require simpler to digest foods, often the byproducts of fungal decay, and often only after such food has been broken or opened up by fungi and others. Compared to fungi, bacteria are in the minor leagues of decaying ability. Fungal feeding. The acidic digestive substances produced by fungi and leaked out of their hyphal tips are similar to those utilized by humans. Fungi don't require a stomach as a vessel in which to digest food. However, like bacteria, fungi lack mouthparts. Instead, fungal decay breaks up organic materials into compounds the fungus can then ingest through its cell walls via diffusion, osmosis, and active transport. Nutrients taken in by fungi are usually immobilized, just as they are when ingested by bacteria and later released. Like bacteria, then, fungi should be viewed as living containers for fertilizer. Excess acids, enzymes, and wastes are left behind as the fungus continues to grow. And as a consequence, the digestion of organics continues even though the fungus has moved on, opening up organic material for bacterial decay and making nutrients available to plants and others in the soil community. Hyphal growth gives a fungus the ability to move relatively long distances to food sources instead of waiting for its food to come close. Though it can clearly do this too, as the nematode-trapping fungus proves. Fungi can, for example, extend up into the leaf litter of the surface of the soil, decay leaves, and then bring the nutrients back down into the root zone, a huge advantage over bacteria, the other primary nutrient recycler in the soil food web. Soil fungi are usually branched and quite capable of gathering organic compounds from different sources simultaneously. Once the nutrient material is inside the cellular membrane, it is transported back through the network of fungal hyphae that often ends at the root of the plant, where some fungi trade for exudates. Thus, the same fungus can extend hyphae downward and outward, absorbing several crucial nutrients: phosphorus, copper, zinc, iron, nitrogen, as well as water. In the case of phosphorus, for example, the propensity of fungi to gather and transport it over distances is truly remarkable. This mineral is almost always chemically locked up in soils. Even when it is applied as fertilizer, phosphorus becomes unavailable to plants within seconds. Not only do fungi seek out this necessary plant nutrient, but they have the ability to free it from its chemical and physical bonds. Yo.
Yo, good morning.
Morning. They then transport their quarry back to plant roots, where the phosphorus is absorbed and utilized. Hey.
Cool.
Oh my. Don't forget that in those instances where a fungus brings food back to a plant root tip, it was attracted to that plant by the plant's exudates. Fungi are good, but the plant is in control. Fungi and plant available nitrogen. Some fungi trade nutrients for exudates, but most often nutrients are released as waste after they are consumed by fungi, or when the fungi die and are decayed. Much of what is released is nitrogen. A key tenet of gardening with the soil food web is that plants can take up nitrogen in two forms, either as ammonium ions or as nitrate ions. The nitrogen released by fungi is ammonium form. If nitrifying bacteria are present, this is converted in two steps to nitrate. The enzymes produced by fungi are decidedly acidic and lower the pH. Remember that bacterial slime raises soil pH. Nitrogen-fixing bacteria generally require a pH above 7. As soils become dominated by fungi, the populations of nitrogen-fixing bacteria required to convert ammonium into nitrates diminish because the pH is lowered by the acids the fungi produce. More ammonium, therefore, remains as plant-available ammonium instead of being converted to nitrates. This has an important implication to gardening with a soil food web. Fungally dominated soils tend to have nitrogen in ammonium form. This is great if you are a plant that prefers ammonium to nitrate, but not so good if you prefer to have your ammonium converted to nitrates. Who wants what is explained in chapter 13? Oh. Fungal adaptations. Fungi have developed all sorts of clever strategies to make it through life. Our nematode strangling fungus proves it. The fungus that developed this very artful and useful adaptation is Arthrobotrys dactyloids. Arthrobotrys dactyloids. The ring that trapped the nematode is actually just a hyphal branch, twisted back on itself. These branches each consist of only 3 cells, which, when touched, produce a signal to let water in. These branches each consist of only 3 cells, which, when touched, produce a signal to let water in. The cells then swell to 3 times their size, and the unsuspecting victim is killed in 1/10 of a second. Pretty amazing. A sophisticated trapping mechanism developed from an inverted branch using only 3 cells. Once again, nanotechnology can only hope to duplicate such a complicated process. Not only does the fungus figure out a way to kill nematodes, which are all blind, but it attracts them to its trap in the first instance. In this case, the fungus released a chemical that attracts the worm. Within a matter of only a few minutes after trapping, the tip of the fungal hypha enters the nematode's body, secretes its powerful enzymes, and starts absorbing nutrients. As this is exactly what the nematode has been doing, eating. The worm is usually a real treasure trove of nutrients for the fungus. These nutrients, of course, are then locked up inside the fungus until the fungus is eaten by one of its predators, or it trades them for exudates. Then the nutrients are mineralized and again are available to plants. The fungus Pleurotus austriatus, the common oyster mushroom you can buy at the supermarket, uses another clever technique to trap food. It emits toxic drops from the tips of its hyphae. An unsuspecting nematode, our perennial fungal fall guy, out and about, looking for food, touches a drop with its mouth, and within minutes it's immobilized. A few hours later, and the fungus is inside the nematode, already digesting it. This is not a bad way to ensure a meal, attract your food, and either trap it or stun it, and then consume it. Other mechanisms have evolved as well. Some fungi use adhesives to stick to nematodes. Other soil fungi trap protozoa and even springtails, much larger microarthropods that are big enough to see with the naked eye. That's fucking nuts. Once attached, the fungi digest their prey and again lock up or immobilize plant nutrients. What drives soil fungi in the direction of particular nutrients is still an open question. It is known that some send out filaments as if they were scouts looking for nutrients. If you have ever seen a well trained bird dog look for a downed bird, you get the idea. The dog circles until its nose finds the bird. Some fungi clearly possess tactile or contact sensing capabilities that allow them to orient in a certain direction so that they can invade their prey or other food source. Sorry, let me read that again. Some fungi clearly possess tactile or contact sensing capabilities that allow them to orient in a certain direction so they can invade their prey or other food source. Others demonstrate the ability to track specific chemicals they know to be in the vicinity of their prey. For the gardener, it is sufficient to know that fungi can find nutrients. When a source is found, fungal strands head over to the area and literally settle in, digesting the material, often combining one nutrient source material with another and transporting nutrients back to the base of the fungus. All the while, other strands scout for more food to attack. Nutrients are held inside the cell walls, preventing them from leaching away. That's pretty crazy. So in the beginning of the book, it displays a couple photos of nematodes doing stuff.
Thank you.
There's a picture of a nematode being wrangled by a fungi, and then another one entering the root of a tomato hair.
Oh, thank you.
One of them, one of them was grown in an environment where it was essentially defenseless, and the nematode was just entering the plant because there was nothing to stop it. And the one that's wrangled by the fungus is what it was describing, how it attacks them. The different fungi have different means to attack their prey with the hyphal tips. Some of them use them pretty much like I've just been picturing, like little tentacles with spears. Jaw! And some of them, I guess, like this one that I just described, was the oyster. Oh gosh, sorry, the actual name of it. Yeah. The Pleurotus austriatus, the oyster mushroom. How I was talking about how that one was consuming its prey by essentially releasing this fluid through its hyphal tips that the prey would consume, be paralyzed, immobilized by, and then it would just digest it from there on. Oh gosh, microorganisms are crazy. Like, could you imagine if this was how we dealt? Just you had that where somebody would just be able to absorb you, or a colony. You go down the wrong street and bam, wham blam jam, just sucked on in, and you're just like the blob. Oh god, it's horrible. Sorry. Fungi and symbiosis. Soil fungi also form extremely important mutual relationships with plants. The first is the association of certain fungi with green algae, which results in the formation of lichens. In this symbiotic relationship, the fungus gets food from the alga, which utilizes its photosynthetic powers, while the fungal strands make up the thallus, or body, of the lichen, in which the pair lives. So the lichen is a combination of fungi and alga? Oh. Like the jellyfish that they thought were like creatures, but it was like a bunch of single-cell organisms and link in the ocean. Oh. Chemicals secreted by the fungus break down the rock and wood upon which the lichens grow. This creates minerals and nutrients for the soil microbes, and plants. The second are mycorrhizae, from the Greek for fungus root, symbiotic associations between plant roots and fungi. In return for exudates from plant roots, mycorrhizal fungi seek out water and nutrients and then bring them back to the plant. The plant becomes dependent on the fungi, and the fungi, in turn, cannot live without the plant's exudates. It is a wonderful world indeed. Mycorrhizae have been known since 1885, when German scientist Albert Bernhard Frank compared pines grown in sterilized soil to those grown in sterilized soil inoculated with forest fungi. The seedlings in the inoculated soil grew faster and much larger than those in the sterilized soil. Yet, it was only in the 1990s that the terms mycorrhiza, the symbiotic root fungus relationship, plural mycorrhizae, and mycorrhizal, its associated adjective, started to creep into the agricultural industry's lexicon, much less the home gardeners. Hey man, thank you for that. Really appreciate the support, Else.
All crew, please proceed to the escape wing for evacuation. This is not a drill.
Hope you're having a good morning. We're the first to admit that we were blindsided by the subject, and one of us had written a popular garden column every week for 30 years and never once mentioned them out of sheer ignorance. A state shared with most gardeners. We now know the extent of our ignorance. At least 90% of all plants form mycorrhizae, and the percentage is probably 95% and even higher. What is worse, we learned that these relationships began some 450 million years ago with terrestrial plant evolution. Plants started growing on the Earth's surface only after fungi entered into relationships with aquatic plants. Let me reread that. What is worse, we learned that these relationships began some 450 million years ago with terrestrial plant evolution. Plants started growing on the Earth's surface only after fungi entered into relationships with aquatic plants. Without mycorrhizal fungi, plants do not obtain the quantities and kinds of nutrients needed to perform at their best. We must alter our gardening practices so as not to kill these crucial beneficial fungi. Yeah. Oh, you got in some Osu? Very nice. Yeah. Oh, yeah. Do you think that's part of that symbiotic relationship with the plants and stuff? I mean, like, the bacteria and the fungi with the plants. So you got plants that are bearing crop or the seeds and stuff, The idea that the plant can reproduce seeds to regrow itself, but not only that, that decomposing organic matter can then be consumed by the bacteria and the fungi. Yeah, about the oh gosh, yeah, it was mentioning earlier. It was like saying that the fungi is totally okay eating the same stuff as the bacteria. It's just that the bacteria is quicker to grab that food and consume it, and it just is able to go and grab other things. It can penetrate cellulose. It was talking about how it could break down lignin, other harder materials that bacteria can't break down, and so it just goes for that instead, right? Oh yeah, we know all about that. 6 Ways Mushrooms Can Save the World by Paul Stamets is a wonderful discussion. A brief discussion on a fucking glimmer of the ideas that guy shares. Yeah. Where am I? Hold on. I lost my spot. Yeah. Perhaps gardeners lack appreciation for fungi because all soil fungi are very fragile. Too much compaction of soil, and fungal tubes are crushed, and the fungi killed. Clearly, fungicides, but also pesticides, inorganic fertilizer, and physical alteration of the soil, rototilling, double digging, destroy fungal hyphae. Chemicals do so by sucking the cytoplasm out of the fungal body. Rototilling simply breaks up the hyphae. The fruiting bodies of mycorrhizal fungi even decrease when fungi are exposed to air pollution, particularly that containing nitrogenous substances. Mycorrhizal fungi are of two kinds. The first, ectomycorrhizal fungi, growing close to the surface of roots and can form webs around them. Ectomycorrhizal fungi associate with ectomycorrhizal fungi associate with hardwoods and conifers. The second are endomycorrhizal fungi. These penetrate and grow inside roots as well as extend outward into the soil. Endomycorrhizal fungi are preferred by most vegetables, annuals, grasses, shrubs, perennials, and softwood trees. Both types of mycorrhizal fungi can extend the reach as well as the surface area of plant roots, the effective surface of a tree's roots. For example, can be increased by a fantastic 700 to 1,000 times by the association. Let me reread that because I kind of just trailed off. Both types of mycorrhizal fungi can extend the reach as well as the surface area of plant roots. The effective surface area of a tree's roots, for example, can be increased a fantastic 700 to 1,000 times by the association. Mycorrhizal fungi get the carbohydrates they need from the host plant's exudates and use that energy to extend out into the soil, pumping moisture and mining nutrients from places the plant roots alone could not access. These fungi are not lone miners either. They form intricate webs and sometimes carry water and nutrients to the roots of different plants. Not only the one from which they started. It is strange to think of a mycorrhizal fungus in association with one plant helping others at the same time, but this occurs. The Bigfoot micro, they have the myco, and the great white is the ecto. Oh, really? Wow, today I learned. Thank you. So now I know. Like, it's something that, like, it probably was explained to me in the past and I didn't even understand. Very cool. Finding and bringing back the phosphorus that is so critical to plants seems to be a major function of many mycorrhizal fungi. The acids produced by mycorrhizal fungi can unlock, retrieve, and transport chemically locked up phosphorus back to a host plant. Mycorrhizal fungi also free up copper, calcium, magnesium, zinc, and iron for plant use. As always, any nutrient compounds not delivered to the plant roots are locked up in the fungi and are released when the fungi die and are decayed. Endophytes. A third important symbiotic relationship between fungi and plants is formed by endophytes, fungi that live most or all of their lives inside the plant tissue. Endophytic fungi, unlike their underground, root-bound mycorrhizal cousins, have adapted to live in the above-ground structures of plants, in leaves, stems, and bark, though some do grow in root tissue. Most trees and shrubs are infected by not just one, but hundreds of different endophytic fungal species, and all plants tested so far, including ocean and freshwater plants, contain at least one kind of endophytic fungus. The reason these ubiquitous microorganisms escaped our attention for so long is because most thrive within their host asymptomatically. That is, happily living away without harming the host at all, and often doing them real good. Scientists have known endophytes since at least 1904, when a paper discussing an endophytic fungus of an annual grass was published. They came into their own in the mid 1970s, however, when a comparison was made of cattle in different pastures. Some cattle were affected in adverse ways by eating grass that have been invaded by endophytic fungi. Attempts to grow the grass in question without the endophyte turned up some surprising things. First, the grass did not do as well. Second, the grass without the fungi lost resistance to its major pests. Oh, damn. A study in the 1980s of ryegrass showed how endophytic fungi greatly increased the rye's resistance to insects. Later studies demonstrated that some turfgrass have an endophyte-derived resistance to other fungi, such as those that cause dollar spot disease, and that there are exceptions to the benign effect rule. Some endophytes cause plants to limit seed production, for example. Oh, damn. For endophytic fungi are Ascomycetes, also called sac fungi, because their spores are produced in sac-like structures. Morels, truffles, brewers, and baker's yeast. And most of the fungi that form lichen are sac fungi, though not all are endophytic. Spores of endophytic fungi are usually transported to their host by way of air currents, having been dispersed from fungi hosted by another plant. Some endophytic fungi invade host seed and grow with the plant. Some insects transport spores as they travel from plant to plant. Cab. Once germinated, the fungus spends its life in the host. As with mycorrhizal fungi, some endophytic fungi require specific plant hosts. Others are much less specific and will infect many different kinds of plants. While endophytic to one plant, the same fungus may be pathogenic to another plant. Several fungi are endophytic in some grasses, but parasitic in others. It appears most endophytic fungi convey some benefit to their host plants. For example, some produce toxins that kill aphids, and others sucking insects, which attack the host and can even stop grazing by mammals. Let me reread that. I'm sorry. For example, some produce toxins that kill aphids, and other sucking insects, which attack the host and can even stop grazing by mammals. Some endophytes improve the host seed germination, ensuring species survival. Others produce antipathogenic substances or induce the host plant to increase resistance to diseases on its own. Many endophytic fungi start the decay process as soon as the host plant dies, ensuring the cycling of nutrients, presumably, to the host plant's progeny, and those of the fungi as well. There is a race to commercialize many of these benefits, promising a whole new set of biocontrols and bioproducts to enhance plant growth, production, and health the soil food webway. Pathogenic and parasitic fungi. Beneficial fungi compete for nutrients and form protective webs and nets, often in conjunction with bacteria, around roots, and even on leaf surfaces, as leaves produce exudates that attract bacteria and fungi as well. This prevents some of their pathogenic and parasitic fungal cousins from invading the plant. The list of fungal pathogens impacting agricultural and horticultural crop is long. The topic fills many books and is beyond the scope of this one. Smut fungi, for example, impact the flour of cereal grains. Rust fungi cause diseases on wheat, oats, rye, fruits, and pines. More common garden problems are downy mildew, root rots, and white rusts. Be there a gardener who has not encountered botrytis or powdery mildew, a catch-all name for a group of fungi that infects different plants with the same results, an unsightly gray or white powdery fungal growth that covers leaves, stems, and flowers. Most powdery mildew, most powdery mildew fungi produce airborne spores that do not require free water to germinate. Given temperatures between 60 and 80 degrees Fahrenheit, 15 and 27 degrees Celsius, and high humidity, these spores germinate and infect their host in your yard. How about Fusarium wilt on tomatoes? The first thing to host in your yard. Oh gosh. How about Fusarium wilt on tomatoes? The first thing to suspect when a tomato's leaves start to yellow when from the bottom of the plant up. It is caused by Fusarium oxysporum, a soil-borne fungus that can survive for a decade or more in dormant stages. It enters plants through roots and invades its water distribution network. Further testament to the power of fungi is Armillaria melia, oak root fungus, which causes oak death, a tiny fungus taking down towering oaks. The fungal activity decays a tree's lignin and cellulose to such an extent that the tree dies. Pathogenic and parasitic fungi make use of various entry points into plants, including stomata, the openings of leaf surfaces that allow plants to breathe, and wounds. And of course, with all this talk of enzymes decaying tough to digest lignin, it shouldn't surprise any gardener that some fungi can dissolve the cuticle and cell walls of the plant it is attacking. If you think this is difficult, think about the fungi that penetrate bathroom tile, and know that some fungi can penetrate granite in search of food. This entire book could be filled with descriptions of fungi that get their nutrition at the expense of living plants. This is not our purpose, only that you realize that soil is loaded with fungi, a concept most gardeners readily grasp because of direct experience. Functional overlap with bacteria. It should be obvious by now that in a healthy soil food web, fungi and bacteria shoulder much the same work and share many of the same functions. Like bacteria, some fungi produce vitamins and antibiotics that kill pathogens in the soil, as well as in the human body. Remember penicillin, the most famous fungus turned antibiotic of all? In 1928, when English bacteriologist Alexander Fleming returned to his lab after his vacation, he found a fungus had contaminated a petri dish full of Staphylococcus bacteria. It ruined his experiment, but no bacteria were found growing near the fungus, and the world of medicine has never been the same. Fungi, like bacteria, play crucial roles in the soil food web as decomposers, nutrient cyclers, soil structure builders, and beneficial symbionts, preventing as well as causing diseases. As well as their ability to impact soil pH, makes them an important tool for gardening with the soil food web. There we go. Chapter 5. Said and done.